next talk is by Dr. Manjunath Sidramaiya, Department of Discovery, Diagnostics and Genomics, Hyclones Life Sciences Solution India Private Limited.
Thanks, everybody. It's indeed a privilege and honor for us, for Cytiva, to present our offering for such a knowledgeable crowd. So today I'm presenting Viax Doctor tissue disintegrator instrument from Cytiva. So it's a tissue disintegrator system meant to disintegrate the human and animal tissue from tissue to single cells. So this is an agenda with what I'm going to talk, overview of single cell genomics and trends and challenges in single cell genomics and sequencing, and single cell sequencing workflow, and some of the data to address the challenge of single cell sequencing or omics applications, and effect of tissue disintegration and the cellular content to retain the cellular content. So how the instrument helps. So and finally the summary. So. Single cell genomics is, single cell RNA sequencing is one of the applications what I'm going to talk, but there are many other omics applications I'm going to take you through. But before going to the single cell sequencing, so I'll just give you the market trend. So if it is forecasted that by 2025, so the market is going to go till 4 billion. So the tissue processing itself in the single cell application is expected to grow to 32 million by 2024. So as I told you, so I'm not only that what we are targeting. We are, in case if you are struggling to extract single cell RNA for single cell RNA sequencing, so this is the talk for you. If you are looking to extract viable cells and good yield of viable cells, so this is for you. And if you are looking for if you want to isolate the cell which represent the original content of the cells, when I said viable cell population, I have the data. It is very clear that when you disintegrate the tissue either by normal regular homogenizer or through manual methods, there is a huge variation in number of viable cells you can recover. So this instrument helps you for that, to recover highest number of viable cells. And when I say isolate the cell which represent the original content, so when you do the disintegration, so a lot of pressure is applied on the cells, either through temperature or or through pressure. Mechanically you put the pressure to disintegrate the tissue into single cells, and that pressure induces a lot of stress-related genes and upregulation of these genes, which you can easily see in your stress-related genes data. So that is actually not because of the true or the microenvironment of the tissue. It is because of the process-induced stress. So to overcome from that, so what are the solutions? So I'm going to discuss that. And in case if you're looking for quick, multiplexable, automated, and high throughput tissue disintegrator, and this is for you. And if you're looking for GMP certified tissue disintegrator, where after disintegration of the tissue, the cells, if you want to extrapolate this for the establishment of the cell lines, and for diagnostics application or for the therapeutic application, this is for you. And if you are looking for control rate freezer, so importance of control rate freezer is, so instead of just throwing yourself directly after disintegration into minus 80 or liquid nitrogen freezing, which puts again temperature related heat shock, to prevent this, you can acclimatize these cells to the particular temperature, and then you can transfer these cells to particular devices. So in case if you are related to any of this application, and this is for you, and I request you to hold on with me for 10 minutes, next 10 minutes. So this instrument, as I told you, is designed for disintegration of human and animal solid tissue into single cells with optimized viability and yield, as well as less stress and burden on the cells. So it is high throughput single cell omics research instrument. It does mild processing, which maximizes the viability and preserves the cell content. And it is semi-automated system, so there is very minimal interaction between the user as well as the samples. In case if you are handling any infectious samples, so you can easily work without coming in contact with the sample as well. So it comes with, as I told you, a control rate freezer, which is you can modulate the temperature of change in the temperature for every minute, so that you can acclimatize your tissue and cells before you put into any temperature based tissue disintegration. So these are some of the instrument components. It comes with wire freeze. So that is where you have digital screen and you can set up your protocols and completely regulate the instrument. And the small box kind of a structure, that is wire extractor. That's where exactly the tissue disintegration happens. And next is omics pouch. So where we put the tissue, and then this omics pouch along with the omics clamp at the end is installed in the omics wire extractor, and this entire wire extractor sits in the wire freeze like this. And the digital screen is provided for setting up your protocol and handling the systems. And this is how the wire extractor, inside the wire extractor, it looks like. It works on the principle of both pressure as well as enzymes. So we have cold enzymes, which operates at 2 to 4 degrees Celsius. And this pressure, the paddles, you can see that inserts the pressure, and we can control the pressure as well, how much pressure is required based on the volume and the toughness of the tissue. So let me take you through the results. So whatever we're climbing, so exactly the result is reflecting. So we have compared it with the competitor instruments as well. So this is the protocol followed, the 35 minutes, 200 RPM and 37 degrees Celsius. And we have used this, only the difference over here is the competitor instrument and the wire extractor. Even enzyme kit we have used from the competition. So to keep it only single variable, and this is how it looks after disintegration. So and passing through the 70 micrometer cell strainer. So if you see, usually it's very common to get 60 to 65% or 70% viability, and so wire extractor has performed better in terms of viability as well. We have got more than 80% viability, and in terms of number of viable cells per mg of tissue also, it is almost double as compared to any regular tissue disintegrators. So I have tissue comparison with the manual method as well. So for the mouse lung disintegration. So again, so we have used in-house enzyme systems, so from the customers, and the manual versus wire extractor tissue disintegration. So this you can easily see that even disintegration is not complete when you do it through manual method. When you see the data, so manual maximum you can get 60% viability, around 60%, and here we have more than 80% viability. And when you see number of viable cells per mg of tissue, it is more than 25 to 30 times better as compared to manual method. I mean, you being a clinician, you people know how difficult it is to get the tissues, clinical tissues. If they don't make use of the tissue properly, if they waste the cells, if these cells are not used properly, so it's a big crime, I would say, because a lot of technical skills, emotions, pain, money, everything is involved in getting the clinical tissue. So this instrument helps you to make use of this tissue in the best possible way. So similarly, I have mouse kidney disintegration. The entire protocol happens in 10 minutes, 200 RPM, and it is compared with manual method again. So in-house enzyme systems, we used it at customer place. You can see a lot of undigested tissue in manual method, so which is completely digested using this system. And again, the results always better whenever we are comparing with viability as well as yield with manual method. So it is more than 5 times better in terms of yield as well here. So second thing I was stressing, to retain the original content of the cells. That this is very, very important with respect to the system. Most of the researchers don't take care of these particular steps. They do the disintegration and then they go for the genomic analysis, metabolomic and proteomic analysis. But what they see is a lot of stress in the data. So either they go for the gene which are involved in the stress, either proteomics or metabolomics. So it looks like the stress-related metabolic the pathways are activated. But those pathways may not be due to exact microenvironment of the tissue, but it may be due to the process where you're putting a lot of stress. So let me show the data. So for that, we have selected 2 sets of sample in triplicates, and we have used common enzyme system for that, and the protocol is here. And again, I'll not go in detail of the viability and the yield. It is very clear that it gives double the viable number of viable cells, and even the viability percentage is also far better as compared to normal tissue disintegrator. But if you see the small area where we have pointed out, red colors, that is only the one a cluster where via extractor has less number of cells as compared to normal tissue disintegrator. So that we just looked into the stress-related genes expression. Most of these stress-related genes are highly regulated, upregulated in that particular population, which is very clear that whenever you use this rough methods or normal tissue disintegrator, most of the genes related to stress are upregulated, and those population have shifted into certain cell clusters which we have taken and we have mapped. And I have one more data. So where we have cold enzymes, this mechanical stress is one type of stress, and the second type of stress is temperature. So we have taken care of that also. So we are using cold dissociation enzymes. This entire system has the capacity to moderate and operate between plus 40 to minus 100 degrees Celsius. So you can use it as a freezer as well and control rate freezer as well. So since it has that capacity, so we have designed a cold enzymes which works at 2 to 4 degrees Celsius for disintegration of your tissue. So where your cell is completely hibernated, the physiological process of the cells are hibernated. So you can disintegrate the cells without worrying about loss of biomolecules or change of activity, a loss of, we're expecting more activity inside the cells. So this is again, so we did it in triplicates. We used our own system, but two enzymes, 37 degrees Celsius enzyme, as well as cold enzymes we have designed. So the genomic analysis again looks like this. So sorry, last 2 minutes, sir, I'm going to finish now. So genomic analysis looks like this. So again, so stress-related genes are upregulated whenever they use 37 degrees Celsius as compared to 2 degrees Celsius enzymes. So it is very clear in both the cases, both mechanical as well as temperature-related stress activities are being taken care during disintegration process. And with that, you can see the viability is more than 90 to 94%, and the yield is more than 6 to 8 million cells per mg of tissue. So which is almost double than what we expect at 37 degrees Celsius dissociation. So this is, it operates at low temperature, less cellular stress, and high viability. In summary, so it is designed for less variation in the samples, saves time, gives you optimized viability, and retains the original content of the cells, and it is flexible to operate with any kind of tissues. Thank you so much.